Hi everyone, welcome to Pure Biology, and today I come with another video that is related to immune system. Obviously, this is today's topic is ELISA or enzyme linked immunosorbent. Clear? And in my previous video, I have already explained about the RIA or radio immunoassay. So you all obviously check out that one. Okay. And this is the here I try to describe about three types of ELISA and with a rough diagram. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and I request you just to the obviously newcomers, please share my channel with your friends if this is helpful to you. Okay, so obviously try to subscribe for the growth of this channel. Okay, so let's continue. This is the ELISA. ELISA is what? ELISA is the enzyme linked immunosorbent assay. Okay, this is the same type of technique just related to RIA, but here few differences are there. What are the difference? That is in here, okay, before going to the description, there are three types of ELISA. I try to describe here. Okay. First one is the indirect ELISA, second one is the sandwich ELISA, and the third one is the competitive ELISA. The first one, indirect ELISA, here the serum or some other sample that containing primary antibody or the short form, that is AB1, is added to an antigen coated microtiter well, okay, and allowed to react with the antigen attached to the well. So here in the first case, serum with sample containing primary antibody, and here I just have denoted AB1. This is added to an microtiter well that is already containing some antigens. Okay, and now we just allow to react with the antigen with the primary antibody. Okay, and here obviously antigen antibody complex are formed. Okay, and after formation of antigen antibody complexes, the free antibodies must be released from the titer well. Okay, so washed away the presence of AB1, then bound to the antigen is detected by, how the bound of antigen detected by? Adding antigen conjugated secondary anti-isotype AB2. Okay, there are in the microtiter well already AB-Ag complexes are there. Okay, and after that the washing of the free antibodies, we have to add what is that? Enzyme conjugated secondary anti-isotype AB, antibody obviously, and this is denoted by AB2. So what must be added there? Enzyme conjugated secondary antibodies. Okay, so this is the adding of this AB2 that bind to the primary AB1. Okay, because these are the anti-isotype antibody. So they not bound to the antigen, they bound to the primary antibody. Okay, and now the free antibodies washed away, and some substrate that is related to the enzyme, obviously this is added, and then a colorful reaction is formed, and this is the product is measured. This colored reaction is occurring, and the product is measured by the specialized spectrophotometric plate readers. Clear? So here the amount is obviously, okay, amount is the color reaction product is measured. So what is happening here? First antibody which are known as primary antibody. These are added to the microtiter well which has already some antigens. Sorry. Okay, so what I'm saying that the primary antibody that is added in the microtiter well which is already containing some antigens. So what is happening there? Antigen antibody complexes are formed, and after formation of the complex the free antibodies must be released, and after that some enzyme coated antibody, that is the secondary antibody and obviously anti-isotype of the primary antibody, okay, must be anti-isotype which are denoted as the secondary antibody. These are added in the medium, okay, or the you can say the microtiter well. And what is happening there? The secondary antibody, these are bound to the primary antibody, and after that some substrate that is related to the particular enzyme already coated with the secondary antibody is added there. So colorful reaction, or you can say the colorful product is formed, and the amount of colorful product is totally determined by or measured by the spectrophotometric plate readers. So this is very important process, and this is the very easiest you can say, and this is very simple method by which we can easily identify the foreign particles that is infected within the body, okay. And here generally HIV can be detected by the method within 6 weeks, okay, within 6 weeks. Next one is the, so you can say this method is very accurate method, and we can easily identify the proper foreign particle that is infected in our body, clear? So next one is the sandwich ELISA. What is happening here? And here the AB1, first AB1, this is immobilized and this is already present in the microtiter well, and then a sample containing antigen is added and allowed to react with the AB, or you can say the primary antibody. After the well is washed, the second enzyme linked specific antibody specific for a different epitope on the AG is added and allowed to react with bound antigen, okay. So what happening here? Here in the microtiter well there is already primary antibody present, and after that the sample containing antigen is added. So what happening there? Antigen antibody complexes are formed, and now in the same method the free antibodies or free antigens that must be I think free antibody, okay, I think free antibody, this must be washed, okay, this must be washed, and then secondary antibody with proper enzyme is added here, and here the antibody is specific for the antigen that is already bound with the primary antibody. Is it clear? Secondary antibody is very specific to bind with that antigen that is already bound with the primary antibody, and obviously secondary antibody containing the enzyme. So next some substrate that is related to enzyme obviously added within that after washing the secondary free antibodies. Then what will happen? Same method, color process or color reaction is occurring, and the amount is just detected, or the amount of the color product is detected, okay. This is the second method, that the sandwich ELISA, and the third method, this is the competitive ELISA. Here the first antibody, this is incubated in solution with sample containing AG, containing antigen, and the complex of AGAB added in microtiter well, which is already AG coated. AG coated means the microtiter well is AG coated, clear, antigen coated. more antigen present in the sample. Within the sample, the solution is taken here, sample containing more antigen. Addition of an enzyme conjugated secondary antibody specific for the isotype of the primary antibody, and that can be used to determine the primary antibody bound to the well. Okay, what is saying here? That already AB is incubated, that means AB-AG complex already has formed. Okay, and this complex is added to the microtiter well that containing already the sample antigens. Okay, and here the number of antigens is greater than the antibody. So after that, enzyme is added here that containing obviously secondary antibody, and that is specific for the isotype of the primary antibody. Okay, same thing, but here is that the AG-AB already formed. After formation of AG-AB, this is added in the microtiter well that containing AG. So here number of AG is greater than the AB. That means the primary antibody. And in the second case, the secondary antibody obviously containing enzyme, this is added here, and this secondary antibody, these are allowed to bind with the primary antibody. Okay, and so here it is easily determined that primary antibody, number of primary antibody bound to the well. Okay, so these are the three types of ELISA. I can explain here. Sorry, I haven't explained here. And just draw. I will try to draw a very rough image of three types of ELISA, so you can easily understand the process. Okay, so give me some time. Okay, so here I just draw very rough structure of the indirect ELISA method, sandwich ELISA method, and competitive ELISA method. Okay, so I have already explained here. So in the next topic, in the next video, I will try to explain another topic. Okay, so you just continue with my channel. Thank you.